Okay, great. So our group is supposed to talk about characterizing functional consequences of genetic variation. So as you might expect, there's some amount of overlap with the topics that were just covered, but I think we've got some other aspects that are quite distinctive. So in thinking about this area, we decided to split the topic up into what we saw as 5 main subtopics. And so this slide briefly summarizes what those are, and then each of the 3 panelists are going to spend a little bit of time going through these. So the first kind of division that we made was to think about the ways that genetic variation is affecting function first in cis, and then in trans, and then how those effects sort of flow outside of cells to affect development and organism-level phenotypes and so on. And then a slightly orthogonal kind of topic direction is in number 2, and this is an idea that Jay talked about a little bit in his overview talk right at the beginning, which is this idea of the Manhattan Project of gene functions. What does every gene in the genome do? And then the last topic, number 5 on there, is to understand how genetic variation acts in the context of our environment. And so we'll talk about that last. And one thing that I want to emphasize is that on the slides, we've focused in large part on kinds of data that we could collect and some of the big questions. But I think it goes, well, it's important to say, of course, that in all of these areas we've got there's an important need for technology development and for data production, but in particular, although we don't have very much in detail on the slides about this, data analysis and new computational methods for interpretation are absolutely essential for all of these areas. So let me hand it over to you, and Neville's going to go first.
Sure.